talking about DNA. And we've talked a little bit about this throughout the year already, but we are now focusing on DNA specifically so we can move into things like protein synthesis and start talking about RNA. So first, where is DNA found? Well, it is found everywhere. Every living thing has DNA, from our bacteriophages, which is shown here, to more bacteria, to our plants, our flowers, even Paris Hilton has DNA. So everything that we have, or everything we know, has DNA. Now, what is DNA? And again, we've talked about it a little bit throughout the year, but DNA is a molecule that contains all of an organism's genetic material and is therefore responsible for heredity. So we talked already about heredity and what gets passed down from generation to generation, and DNA, those genes that lie basically right there, that is what is responsible for everything to do with an organism. So a strand of DNA contains many genes, all of which code for proteins. So this is what differs a little bit from what we've previously talked about. So we've talked about the genes and how genes can give us a phenotype, basically what we look like or what blood type we have. But these genes actually code for proteins. Those proteins are responsible for what we know today and a lot of our processes. So each of these proteins has a different function in a cell, and we'll talk a little bit more about those as we go forward with this unit. So two scientists actually took credit for discovering the structure of DNA. So around 1960s, we really didn't know what DNA looked like. We did know, however, that DNA kind of existed, but we didn't really know what it looked like or the structure of it. So two scientists, James Watson and Francis Crick, were the first to discover the structure of DNA. Now, this doesn't necessarily mean that they actually did it. Please understand that science has, and a lot of different kinds of people work on a lot of these discoveries. So actually, Rosalind Franklin was the one who discovered DNA, the structure of DNA. However, she isn't fully credited with it because James Watson and Francis Crick were the first ones to actually publish on it. So if you can tell me tomorrow after watching this video, Rosalind Franklin was in fact the true discoverer of the structure of DNA, I will allow you to have 2 extra bonus points on your test. Now, they referred to DNA as a double helix because it contains 2 complementary strands, each of which is shaped like a spiral staircase. So if we were to unwind this DNA, it looks like a ladder, but it's actually wound up. So think of it as one of those spiral staircases that you might see on a boat or in a small house or anything like that. Now, this is just a picture showing you kind of where DNA is located. And so throughout the year, we've talked a little bit about every single part of this. So we've gone from the cell and the nucleus of a cell, and inside the nucleus actually contains our chromosomes, our DNA. And this appears to be a human cell because it says there's 23 pairs of chromosomes. So inside this nucleus, we have the DNA, which has our genes, basically chromosomes, and made up of genes. And those genes, we go a little bit closer and we're starting to see the DNA. And we can see the bases on the DNA. And we're going to talk about those bases here momentarily. So what's the function of DNA? Well, there's 3 big functions that DNA has. The first one is something that we already know, right? The first one is storing information. So DNA stores genetic material or genetic information in genes that control development of an organism. So these genes are factors that are passed from one parental generation to the next, and they're going to determine an organism's characteristics. Now, for most of us, like humans, for example, we get a copy from mom and a copy from dad. Typically, this is how sexual reproduction works. You get one copy from one parent and another copy from another parent. Another function of DNA is just to copy information. So an exact copy must be created before cell division can take place. And we've talked about this when we talked about mitosis. So it's very important for each cell in an organism to have exactly the same copy of DNA. Now, the cell itself, and we'll talk more about this in future units, but the cell itself might have certain genes turned on and off in order for it to do the function that that cell needs to do. Like blood cells, for instance, are going to look a little bit different than, let's say, a skin cell. They might have certain genes turned on and off, so they work how they're supposed to work. But that's basically what the DNA does. It turns things on and off. A third function of DNA is gene expression, and we are familiar with this as well, since we just talked about heredity. So this is the information coded in the DNA that's expressed in an organism. So how is the DNA? How are the genes read in an individual? So what do we physically see? What is it that kind of is expressed in an organism? Now, I mentioned that DNA looks like a winding staircase, so we know that it is in a shape of a double helix, as pictured here in the slide. We know that DNA is made of repeating subunits called nucleotides or nitrogenous bases. This was one of your vocabulary words. And the double helix is composed of two nucleotide chains twisted around a central axis, and they're actually held together by hydrogen bonds. Now, we talked about hydrogen bonds when we talked about water way back in the day. And so these hydrogen bonds are weak bonds. They can be easily broken, but they're strong enough to hold these nucleotides together. So in the picture here, and we'll talk more about this in class, we have these bases here, these nitrogenous bases, held together by these little hydrogen bonds that keep them together, almost like Velcro. So they're pretty strong, but they are pretty weak compared to other types of bonds. Now, DNA stands for deoxyribonucleic acid. Most of us just say DNA because it's a lot easier. But DNA is made up of three main components. So pictured here is a version of DNA. Now, I have already kind of created this little a little ladder for you so you can see. So if we have unwound the DNA, and when I'm scribbling in right here, that's one side of the ladder. And then what's across here, down towards the bottom, is one of the rungs of a ladder. So if we unwind it, it looks exactly like a ladder. So there's three parts to this DNA. The first is deoxyribose. Now this is a sugar molecule, and it looks exactly like a sugar molecule. So if we go back way back when we were doing macromolecules, this would have a similar shape when we were talking about those sugars. So deoxyribose is the name of the sugar that's found in DNA. Ribose is the sugar that's found in RNA, and we'll talk about that in another video. We also have phosphates, and phosphates make up the part of the backbone of the DNA. So remember, that side part of the ladder, the phosphates are important to make up that side part. And then we have these nitrogenous bases, and we know that there are 4 nitrogenous bases for DNA. We have thymine, adenine, guanine, and cytosine, and we're going to talk more about those in the next slide. But if you kind of see here, the nitrogenous bases make up the rungs of the ladder. So phosphates make up the sides, kind of like the backbone, and then we have these nitrogenous bases making up the rungs of the ladder. Now let's talk more specifically about these nucleotides, or as I might call them in class, or as they are known as nitrogenous bases. And the reason why they are actually called nitrogenous bases is because they do have nitrogen. So they're made up of nitrogen. And again, think back when we talked about macromolecules, we were talking about those nucleic acids. Nucleic acids are made up of CHOMP, remember? Carbon, hydrogen, oxygen, nitrogen, and phosphorus. So this is part of that. We just saw the phosphorus part. Now here's everything else. The CHON, if you will. So nucleotides consist of a sugar, a phosphate, and a base. So all three parts of those make up nucleotides. So adenine, thymine, guanine, and cytosine are the actual bases that make up these nucleotides. So we couldn't, a lot of times they are talked about nucleotides versus bases. We talk about them as being the same thing, but in reality they are not. Nucleotides are all three. The bases are just those four: adenine, thymine, guanine, cytosine. So nucleotides pair up in very specific ways, or these bases do. So adenine pairs with thymine, guanine pairs with cytosine. So you can think of it as the adage that apples are in trees, cars are in garages. So you can think of it in terms of that. Now, we'll talk about in class what makes them a little bit different from each other. So you see the structure here, how adenine and guanine, how these guys have basically these two-part structures here, thymine and cytosine. They're only made up of one of these structures. So while not necessarily important to know for our EOC, might be good to know for future biology classes or those you might take in college. So how the code works. The rungs of the ladder can occur in any order, as long as that base pair rule is followed. So as long as A and T and C and G go together. So A, T, C, G, as long as that base pair rule is followed, it's all good. Now, it can occur in any order, and this is based upon the organism, the type of organism it happens to be, what that DNA is trying to read out or code for. So it can really, there's really no specific sequence. Now, of course, for humans, we have some sequence in the same order. Chimpanzees, of course, are the same. So by species, of course, you're going to see some similarities, even across species. So like, for instance, humans are part of the primate family. So we have chimpanzee DNA and we have great ape DNA, very similar to ours. So if a stretch of DNA could be AATGACCAT, so that fancy thing in blue, it would actually code for a different gene than a stretch that might read GGG, So each of these, think of it as a language. So each of them are going to code for a very specific protein, which might be something completely different. And that protein will tell the cell, the body, whatever it happens to be, to do specific things. Now, there are endless combinations with these four bases. So just like letters of the alphabet, they can combine to form many different words. And again, later on in some of these videos, we'll see exactly how this happens and how to code for different proteins.